Hello everyone, and welcome to today's live broadcast, Identifying Genes Related to or Specific for a Disease of Interest, presented by Philip Zimmermann, PhD, the CEO of Nebion AG in Zurich, Switzerland. I am Kristen Peregentiel of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots. Labroots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window. Or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Philip Zimmermann. I will now turn the presentation over to him.
Well, hello everyone. It's a pleasure for me to be here at Labroots today. It's the first time I am giving a presentation, and I'm really excited to be here with Labroots to talk to you about the topic of identifying genes related to or specific for a disease of interest. So let's get started immediately with the topic of differential expression. So as a scientist, you may have to do gene expression experiments where you compare a treatment and a control. And typically you would either have your own experiment or you choose one from GENEVESTIGATOR, and you define the treatment groups and the control group, and then you can run a differential expression analysis. You can do different types of differential expression analysis, like diseased versus healthy, or a treatment against untreated, or a time point A versus a time point B. Or you may compare different doses inside an experiment, etc. Now, in your particular case, you will be working on one scenario. Let's call it disease number 9 in this example. And you will look for genes that are up or down regulated in that disease as compared to healthy tissues. So what you will get in such an analysis is a list of genes that are upregulated or downregulated in your particular disease. Now what you don't know if you do this analysis on its own is what do these genes that you have found do in other diseases. You don't take this into account because they are not part of your analysis. But let's assume you would know what happens to these genes in all of these other conditions. Then you would find something like this, where genes that in your experiment are very strongly regulated are sometimes very specific for your disease. For example, the third one here, where we see no regulation in any of the other diseases, but it's regulated in your disease of interest. But you will also get lots of other genes that are regulated either up or down in other diseases that you may not have been aware of if you have never looked at these data set at the same time. So assuming you would have this possibility to align other studies, other diseases together with your disease of interest, you could see these effects. And then you could start working on changing the order of your results, such as to find genes that are really specific for your disease of interest, as you can see here. We have taken the same plot as here, but we have simply ranked those genes down that are also regulated in all kinds of other conditions, and we keep those that are specific for the disease. And so what you end up with is a list of genes that is specifically regulated in your disease, as opposed to simply being regulated in your disease as compared to the control when it's analyzed on its own. So what does it need to do this type of analysis? The traditional way of doing differential expression analysis is you have your experiment, you compare treatment to control or disease versus healthy, you get a list of genes, and then you interpret it in some way or the other. And the interpretation of this list is always that these genes are somehow related to your disease. Unfortunately, many scientists interpret it as being these are the disease-specific genes. But as we have just looked at before, they are actually not really specific for the disease. They are only in some way related to the disease, or they are the genes that respond as an effect of the disease. But they may not necessarily be specific. So if we want them to be specific, to find the genes that are really specific, we need to include all the other diseases as well. So then we are working on the level of analyzing thousands of experiments, and we can run different types of analysis on it on a compendium-wide level, and also we can interpret them on that level rather than interpreting one single study. Now, in order to have all of these other diseases combined with our disease of interest, we need to integrate all of these experiments. So what we do in our lab here is to curate public expression data that we collect from different sources like Gene Expression Omnibus, European Phenome Genome Archive, ArrayExpress, the Cancer Genome Atlas, and many more. But as you may know, these data sets, although they are available, which is a really great thing for our research community, they are not fully standardized between the different repositories, but also they are available in different formats, in different qualities. There is a big heterogeneity. And so we have a team of curators who work on collecting these experiments, on integrating them into the database, curating all the annotations, which means all the descriptions of all of these samples, and then we bring it all together into a database where everything is completely systematically annotated, described, but also quality controlled and normalized in a way that we can compare between experiments. So once this is done, you have the possibilities to run new types of analysis that you would not be able to do on one single experiment. And in our particular case, we have developed two tools to do that. One is called GENEVESTIGATOR, and the other is called GeneVisible, which is a completely free online tool, which also runs on mobile devices. And it allows you to take advantage of all of these thousands of studies to learn more about your genes of interest, or to find genes that are specific for a disease, as we are looking at in our particular case. Now, just to give some detail about the curation process, which is really important for us. So the first thing we do is to control the quality of the data, which means the statistical quality. We detect duplications that can happen in multiple studies. For example, you have a set of reference samples that is used in two different studies, but they are the same sample. So we can identify such duplications. We check the design of the experiment, and then we read the publications and verify if we have the same design, and also we get information about every sample so that we can annotate and describe them well. We use occasionally also markers to verify some annotations, and of course an important part of it is to make sure that the work done by one curator is peer-reviewed by another curator, so that we can avoid errors that can be caused by human intervention. And the second level is on the level of the metadata. So all these samples need to be described properly, so we need controlled vocabularies that describe them using the same words. And we do that on the level of the experiment, on the level of the samples, and also on the level of the comparisons. So the curators manually, based on the experimental design, can define groups of treatment to control or diseased versus healthy, and they enter that into the database. We also add patient characteristics and the mappings of the different replica into single nodes that have particular properties. So it's quite an intensive work to curate all of this data, but at the end, once everything is available in such a systematic, such a clean and such a high quality state, you can start doing analysis types that you would not be otherwise able to do. So there are three levels of data analysis that you can do with such a high quality compendium. The first level is to pick one experiment out of the database and to simply run the type of analysis that you would traditionally do also on your own experiment, for example, differential expression analysis. The second type is rather new. This is where we have specialized our tools on. It's called platform level. So there we allow to assemble tens or hundreds or thousands even of studies into one big data matrix, and to be able to run queries like which genes are specifically expressed in one disease but not in any of the other diseases, or respond to a particular drug and not to the other drugs, or to a group of drugs that have the same mode of action or whatever. So you can start identifying genes in a much more specific way than if you do them individually on each experiment, and you start to compare. And so I will show you an example afterwards what is the difference between doing meta-analysis on individual experiments and doing a global search across all the data at the same time to find genes that are specific. And then there is a third level, which we will not look at today, but it's about assembling results and not any more measurement data between different studies that you have assembled. And this approach is very flexible because it does not have to go by platform, not even by data type. You can actually bring together anything together that has a list of genes, and you can run enrichment. Good. So let's have a look at one example where we will try to understand what's the difference between running differential expression analysis on individual studies and running a global search to see which genes are specific for the disease. So we're going to look at Crohn's disease. It's an inflammatory disorder that is present at about 3 per mil in the population, and there are 35,000 deaths annually due to this disease. It's a disease that is currently incurable, so we maintain it in remission essentially and prevent relapse. So in this example here, what we are going to do is to compare diseased samples versus healthy samples of colon mucosa tissues, and this comparison was found in 4 different studies. There were actually more, but we randomly chose 4, where we saw from the descriptions that all of them had collected colon mucosa tissues from diseased and from healthy adjacent or healthy patients, and allow you to make such a comparison and see which genes are regulated in response to the disease. So what we did was to try to have approximately 200 genes, except for the first one here, where we did not get quite that high, but we didn't want to have a false discovery rate too bad in that case. So we kept it at below 0.1, but otherwise we are between 150 and 200 genes that are regulated. And what you see here are the plots of differential expression, where we kept only those genes that are upregulated in response to Crohn's disease as compared to the healthy controls. And in the middle here, what you see is a Venn diagram with 4 colors, each color representing one plot. And maybe it's a little bit small to read it, but what is striking here is that the overlaps between the different studies is relatively small. Let's go to the next slide to see what it is. So from 601 genes that are upregulated across all of these 4 studies, only 2 genes were found to be upregulated in all 4 of them. In 3 of the 4 studies there were 14 genes, and in 2 of the 4 studies we found 21 genes, but all the other approximately 560 genes that were found to be regulated in only 1 of the 4 studies. And this is really surprising, because you would actually expect that if a study is well done and the samples harvested are the same disease on the same tissue type against the same healthy controls done on the same tissue type, you would expect to have not 100% overlap, but maybe 30, 40, 50% overlap. And here it's just a few percent, which is close to being a random overlap between lists of this size. Now, if we look at the function of these genes, for example, we take genes that are present in 3 of 4 studies. This is the list of 14 genes that we have, and we enrich it against pathways. In this case, we chose reactome pathways. You can see that the pathways that pop up as being the most significant are related to the immune system. So obviously, the list of genes obtained has something to do with the immune response. It's not really surprising, since Crohn's disease is an autoimmune disease, but it may also mean that the genes that we observe in the different experiments reflect processes that happen in Crohn's disease, but may also happen due to other things, maybe to the microbiome, maybe to other diseases that these patients had, but which we are not aware of. So why do so few genes overlap between the different studies? And this is a phenomenon that we have observed not only in Crohn's disease, but also in several other diseases where you compare diseased samples versus healthy controls. So one hypothesis is that the tissues harvested don't have exact same cell type composition. And the evidence for this is what you see in this plot here on the top left or top right for plants, where we compared the variance that is caused in the transcriptome between a tissue and the average of all tissues on the one hand, and between a given disease and the healthy controls, or a given perturbation and the corresponding controls, which are down in the lower part of the plot. And what you see here is that the difference in transcriptome population is about 10 times higher if you compare any given tissue against the average of all tissues than the effect that you would see if you have a disease against the healthy controls. So comparing different tissue types, you may have differences in genes being expressed of 2,000 to 5,000 genes, whereas between a diseased and a healthy control, if it is well done, you would rather have a few hundred genes that are regulated. So the cell type composition may strongly bias this type of comparison, which means that in different laboratories they may have been collecting different parts of tissues. Some may be collecting only the superficial part of the mucosa, some may be going a little bit deeper, and due to that the composition of the tissues is different. If they have collected differences also between the diseased and the healthy, they will, especially in that case, get also very big differences in expression, which have nothing to do with the disease, but with the composition of the tissues. So this could be one explanation, but there could also be other explanations why there are so big differences between the different studies. So what we try to do here is to answer the question, how can we find genes that are really specific for a particular disease and minimally in the sense that they are strongly regulated in the target disease, but they are minimally regulated in all other diseases and conditions. So we developed an algorithm where we score every gene of the transcriptome in a big data matrix where we have thousands of different comparisons of diseased versus healthy, but also drug treatments against controls or genotypes against other genotypes. Whatever kind of comparison that can occur in transcriptomic studies, they are assembled in a big data matrix, and we have the expression values for all the genes, and then we score all the genes for their specificity of expression in a chosen target category of interest, for example, Crohn's disease, against all the other conditions. Or if you are only interested in comparing with other diseases, you compare only with that section of all of these comparisons. So it's a new approach where you don't work on one individual study, but you assemble thousands of studies and you work on all of them at the same time. Now the reason why this is platform level is that we need to have a data matrix where we are measuring the same genes in the same way so that we have comparable measurements. So unfortunately we cannot mix all kinds of microarrays and RNA sequencing platforms to do that on the one hand, but still, if you have a compendium large enough with several hundred diseases and potentially thousands of different compounds and conditions, you will have an excellent starting point to find genes that are specific for a disease. So in this case here, we have used Affymetrix 133 plus 2 studies. It's a very popular platform, so there are very many studies available, and we looked for genes that are specifically expressed in Crohn's disease. Then we compared the results that we obtained with the genes that were found to be regulated in the 4 Crohn's disease studies that we had analyzed previously. And what you see here is the overlap in the right table. The red dots indicate the genes that were found, the 25 genes that were found to be specifically expressed in Crohn's disease, and the other colors are genes that were found in the other studies. So from the 21 genes that were present in 2 of the 4 studies, only 4 were among those that were found to be Crohn's disease specific by our analysis. From the 14 genes that were present in 3 studies, 4 were found in our list, and from the 2 genes that were present in all 4 studies, as being regulated in all 4 Crohn's disease studies, both of them were found in our list. However, what was interesting was that there were still 15 other genes, which is more than half, which were not found in these overlaps here. Now this is another representation of it. You can see the same bars as we saw before. So the 2 genes that were found to be regulated in all 4 studies, when they are analyzed separately, were also found in our list. But from the 14 that were in 3 or 4 studies, only 4 were in our list, and all the others were not in our list. So they were genes that are somehow responsive in Crohn's disease samples in 3 or 4 studies, but they are not very specific for the disease. They might be genes that are, for example, inflammatory, but they are involved also in many other inflammatory diseases, or immune response genes that are also involved in other processes, which, of course, will pop up if you compare only that disease versus healthy controls, but they will not be scored well if you screen them against all the other diseases. And if we look at what overlaps in 2 or 4 studies, there are even less, and in percentage. And of course, from the many, many genes that were found to be regulated in at least one of the 4 studies, we had 12 of our genes found in there, which represents only 2% of the entire list. Now this is a little bit to illustrate the case of differential expression in one study versus the global search. So the blue box here illustrates the section where we have 25 genes found in this particular gene expression study that was published in GEO from Crohn's disease versus healthy controls. And these are the top 25 genes. And what you can see here is that these genes are very strongly upregulated in response to Crohn's disease. This is actually what we were screening for in the differential expression. We see that they are also strongly upregulated in ulcerative colitis, which is a related disease, so it's not really surprising. They are also, to a certain extent, regulated in colorectal adenomas. And all of them are very strongly downregulated in response to infliximab, which is one of the frequent drugs used to treat Crohn's disease. So there is a clear interaction between the expression in response to the drug and expression in the disease itself. So this is a very nice result that we can see here. We see the same results also in the top 25 genes that were found in our analysis where we screened for genes that are only or maximally expressed in Crohn's disease and minimally expressed elsewhere. But now, since we had been screening for genes that are specific for Crohn's disease, if we add other diseases, as I have added here, these are not all that we tested. It's just an example to illustrate it. But for example, here in these inflammatory diseases, we can see that the genes found to be top regulated, if you do a differential expression in this study, were also regulated in quite a number of other inflammatory diseases. Whereas in our approach, the genes we found were specific for Crohn's disease. They were not regulated in these other inflammatory diseases either. So it allowed us to screen for genes that are really specific for the disease of interest and not involved in other things, which makes them much more interesting or in a certain way or attractive to be studied as novel targets for the disease. Unless you want to develop a drug that can be applied to other inflammatory diseases as well. But if you want to do drug discovery specific for one disease, or you want to study the mechanism of that disease and understand the biology, it might be worth also looking at the approach that we propose here, where you find genes really specific for the disease of interest. And there are also other diseases here, cancers, and I did not include the different drug responses here, but except for infliximab and brodalumab, but otherwise I did not include them, but there are also interesting effects that you can see there, where the genes we found to be best scoring were actually not changing very much in any of those other conditions, but the genes that you find looking in a very focused individual way of disease versus healthy in one single study did not allow to screen against that. Now, if we take these genes that were found in 2 of 4 studies or 3 of 4 studies, and we look at their expression in different tissues, what you see here is an export of the tissue level expression of these 21 genes and 14 genes that we saw before across more than 400 different tissue and cell types. These are figures done with GENEVESTIGATOR. What you can see here is that these genes are sometimes specific for particular tissues, like the ones here on the right of the first figure, but you can see also genes that are expressed in many, many different tissues. If you compare that to the third figure you can see here, this figure here, where we perform the global search of Crohn's disease against all the other diseases, what is interesting is that these genes found were all expressed in tissues that were very relevant for the disease. So they had a very strong expression in colonic mucosa, in also the small intestine, pancreatic duct, the conjunctiva, nasal epithelium, but especially also in extracted colonic mucosa cells and pancreatic islet cells, and also here alveolar sac cells. So these are all cells with some epithelial structures, and the genes that we found to be very specific for Crohn's disease had a very strong and much more specific expression in those tissues that are relevant for our disease than in any of the other tissues, as you would see the case here in the other diseases here, where you have expression in all kinds of other different tissues as well. So that was a nice confirmation for us that the approach we followed not only allowed to find genes that are specific for the disease, but also that we saw they were expressed nicely in tissues that are relevant for the disease. So in summary, the approach that we propose here to the community is, on the one hand, a single experiment analysis where you compare a treatment and a control, or a disease versus healthy, and where you do collection-wide analysis or platform-wide analysis, where you assemble data from hundreds or thousands of different conditions, and then you screen for genes that are specific. And in the first case, it's a very useful approach. We don't want to in any way discredit this approach, but it doesn't answer quite the same question as the one we were trying to answer. So in the first case, you are looking at genes that will be regulated in response to the disease without having information about how they are regulated in response to other diseases, but you know that they are somehow related to your disease of interest, and that is already a very important piece of information. Whereas in our case, we were interested to know which are those exclusively expressed, or if it's not possible, of course the biology may not allow it to be exclusively expressed, they might be related diseases, but at least you will find those genes that are as specific as possible to your disease of interest. And this has maybe different types of applications than the first type of, let's say, traditional differential expression. In this case here, it is very useful for the identification of biomarkers or to identify novel targets that could be used to treat the disease, or if you are studying the biology of the disease, you will come across new genes that you may not have seen before, because these genes that come up as disease-specific are not always those that are the most strongly regulated in terms of fold change or intensity, but they are simply expressed or regulated there, but not elsewhere. And that's why frequently they drop out of the analysis if you do it in a disease versus healthy way on single experiments, because they are sometimes not very strongly regulated, but that's not what we are scoring for. We are scoring here for specificity, and that's why we get only a partial overlap with the results that you get in, let's say, traditional differential expression analysis. So the conclusion is that the differential expression analysis, as we traditionally do it, where we compare one group of diseased versus the healthy controls, allows to find disease-related genes. And for the example we chose, the overlap was actually even very low, which was quite surprising, but that's a fact. And only a small proportion of these disease-related genes were disease-specific, as we could see in the slide here. The overlap between disease-related and disease-specific was relatively small. There were 5 or 6 genes here, which were also found using traditional differential expression, but were found if we did scoring against all other diseases. But it's only a small proportion. All the others were genes that were also regulated in other conditions if we did differential expression on one study at a time. So the compendium-wide search to find genes regulated in a chosen disease versus minimal expression in other diseases allowed us to find disease-specific genes much more efficiently. But it's not something that is so easily done, because you have to assemble all of the data sets, you have to prepare them to be normalized in the same way that they can be compared and analyzed all together. And that's why we developed a tool that allows our users to do exactly this type of analysis. So I hope we have been able to show a little bit the work we have been on in the past years to allow this type of analysis. And if you are interested to run such analysis, you're welcome to contact me or through our website, and we can help you getting this done for your disease of interest. I would like to acknowledge different organizations who have been working with us in the past years. First of all, ETH Zurich, which is the parent university from which our spin-off has started, the Swiss Commission for Technology and Innovation, VentureKick, and of course our software developers and biocurators, who have really done a great job of assembling all of the data and making tools for our scientists to be able to analyze all of these data sets and run new types of queries. And I would like to thank all of you for joining this session, and I'm open to take questions.
Thank you, Dr. Zimmermann, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question that you would like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many questions as we can. Let's get started. Our first question is, why do you think the results from the different Crohn's disease studies don't overlap well?
So I think personally that, as I showed in the slides, that probably the main reason is that the collection of the different biopsies, which is not done in the same way in the different laboratories, where some may be collecting adjacent tissues or the proportions of the different cell types is different. And this is something which we have been thinking a lot about because it causes big biases in the differential expression analysis in many studies. And we are currently working on methodologies to be able to standardize the differences in cell type composition so that we can fish out much more specifically the genes that are really caused by the disease and those that may be caused by differences in composition.
Thank you, Dr. Zimmermann. For our next question, we have, why do we need the entire data sets for all diseases to find genes specifically regulated in a single disease? Wouldn't it be enough to compile the lists of regulated genes for each disease and then search for genes that are found exclusively in the list from the disease of interest?
Yes, that's a good point. There have been different approaches published to date where called meta-analysis, where you collect results that have been produced in different studies looking at the same disease process. And you may collect, let's say, a few hundred genes for every one of these studies, as we did for Crohn's disease, and then you start looking at what overlaps between the different studies. There are methods that go a bit beyond that, where you can assemble all the gene expression measurements for all of these contrasts, and then you run a meta-analysis on it to find genes that are significantly regulated. So not only looking at overlaps of lists, but really doing statistics on this subset of studies. But in that case as well, you are still looking only at what happens in Crohn's disease versus controls, or in any other disease versus the controls. And what we wanted in our case was not to focus only on one disease, but to be able to answer the question, which genes are specific for my disease as compared to any other disease, so that we can get a grasp of what's really happening in that particular disease and what could be the cause of that disease, or where could there be a point of intervention specific for that disease, which we now know that it is not involved in anything else, and it is something new. So that's the reason why we need all the measurements for all the genes, and not only the results that are collected from different studies and doing an enrichment sort of analysis, but we need to be able to screen globally for all the genes and then get out the best ones.
Great. Okay, it looks like we have one more question for now. A disease can be manifested in different tissues. How do you define disease specific in such a case?
Yes, this is a good question. Frequently, when we talk together as scientists, we don't really talk precisely. We say things like, I'm working on genes that are related to a particular disease, and somebody else might be working on the same disease but on another tissue type, for example, on blood samples, whereas the other are working on the diseased organ The third one may be working only on a subportion of the blood, like on immune cells, and so on. And each of them will be, in his or her mindset, will be thinking that they are working on the genes that are involved in the disease. But if you would compare what happens in the different processes, they might be completely different, and so the lists of genes they are working with will be completely different. So it's true that I think scientists, we need to be a bit more aware of the context around which we are working, and maybe formulate it a little bit more precisely, that if we are working on a diseased organ, for example here mucosa tissues of the colon, we can say we are working on genes involved in the disease on a particular tissue type, but they may be different than the person working on a diagnostics to identify markers for the disease in body fluids, which may be a completely different process. I think it's a lot of a question about how precisely we formulate the question of disease genes. So yeah, I think we should be careful not to just generalize and say these are the disease genes for that disease, but we should always put the context to it, in particular the tissue or cell type context on which one is working.
I would like to once again thank Dr. Zimmermann for his presentation. I would also like to thank Labroots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August of 2017. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.